So thank you everyone for joining our first Horizons in Neuro-Oncology for 2025. Just a bit of housekeeping. We will be recording this session so that it could be made available at a later date on our YouTube channel, which we will send out the link for where that can be found at the end. And for those who've never joined a Horizons in Neuro-Oncology before, the normal structure of these sessions are that we will have a speaker, and today we're fortunate enough to have Dr. Sanai, who will give us a 45-minute overview of his work, which is going to be very exciting, and then we will open it up to 15 minutes of Q&A. And we do ask, if you are in the audience, we do allow for 15 minutes of Q&A where we will ask you to put your hands up and do a live Q&A, but please stay on mute, so to avoid any disruption. So as I said briefly, we are very, very lucky to have Dr. Sanai from the Barrow Institute in Phoenix, who is giving up his Tuesday morning in his time to come and speak to us about how he's pioneered the way for window of opportunity trials to phase two in brain tumors. And I will hand over to Dr. Sanai.
Thank you, Lily, and thank you, Matt, for the invitation, and great to see some old friends in the audience. So I'll spend the next 45 minutes, or hopefully a little bit less, discussing what I think should be a more emphasized strategy for drug development for brain tumors, and I'll try to make the case as to why. No relevant disclosures to the topics that are going to be discussed in this lecture. As a reminder of what you already know, the conventional wisdom for drug development is the bench to bedside model, with discovery-based science occurring typically in an in silico, in vitro, or in vivo preclinical phase. And then those hypotheses tested there subsequently being advanced to patients, and typically in the setting of small molecules and other experimental therapies. Those patient studies are phase 1 studies, phase 2, and phase 3. And so this has been sort of the system that has served us well in oncology in general for the last era. But I think what I would submit is that the system has not necessarily served us as well in CNS oncology, which is sort of a self-evident statement. One way to sort of clarify what's happening within our space is to look at the latest phase studies. So these are the phase 3 studies. These are the very large endeavors that require a lot of resource as well as quite a bit of time and patience to complete. And obviously within CNS oncology, there are not nearly as many phase 3s as there are in other oncologic specialties for obvious reasons. But if we look at the ones that have been completed over the last couple decades, it's instructive to understand where the successes and perhaps the failures have originated from. So we know that there have been 3 positive phase 3 studies in the most recent era: the STOOP study with temozolomide, a subsequent study looking at older patients with GBM, and a modified radiotherapy course for them, what we call the hyperfractionated regimen, and then of course Tumor Treating Fields with the Optune device. But if we look at the studies that led up to these phase 3 studies, so the data that were cited as the rationale for entering these 20 randomized phase 3 studies, many of which were global, you'd be surprised to see that few of them actually had what would be considered sort of standard or appropriate levels of phase 2 data. In several of them, there were no phase 2 data whatsoever, and the study was initiated without a phase 2 study. A smaller subset had phase 2 studies that I think were conventionally considered to be inadequately designed, with some overestimation of benefits and some studies that were clearly negative at phase 2 but still advanced to phase 3. And so, this is not to second guess what happened, but rather just to acknowledge the realities of how experimental therapies are advancing in the modern era within CNS neuro-oncology. Now, these are studies that have already been completed. If you look at the ones that are currently ongoing, so studies that have been initiated from 2014 onward, many of which have not been complete, have not been finally reported, we can look in a little bit more detail as what was known about the drugs in these studies. And interestingly, the drug classes are far more varied in recent years than they were in the previous decades, which is a reflection of where drug development is going within oncology in general. But in almost no case were there any real analyses of how the drugs performed, both in terms of CNS penetration, which we call pharmacokinetics, or target modulation, which we call pharmacodynamics, in patients. So a lot of these data were just extrapolated either from preclinical studies or in many cases, they were never extrapolated at all and just simply assumed. Now, from a regulatory standpoint, this is perfectly acceptable because neither the EMA nor the FDA have any predefined standards for what needs to be demonstrated at a PK/PD level for any drug to go into a pivotal study or a registrational phase 3. They simply defer to the companies in terms of their kind of personal private assessments as to whether the drugs have the adequate properties to have a clinical effect. And that may be part of the challenge. But the problem, of course, with that kind of model is that what it means is that in almost all cases, you're relying upon an animal model to inform you as to how some therapy will work in humans. And what we know after decades of experience is that there has never been an animal model study that has accurately predicted the clinical results of a phase 3 study. And this has been the case across any drug class you can imagine, where in all of these examples, there were animal data that clearly demonstrated tumor-related and survival-related effects. And then there were clinical data that absolutely demonstrated no effect. And so this kind of raises this question as to whether or not the reliance on animal studies is sufficient. If we narrow that analysis down to just a single parameter, that single parameter that largely determines the fate of most experimental therapies is the ability for a drug, for example, to penetrate the blood-brain barrier. And the pharmacokinetics of how that works is fairly well understood and governed by a hypothesis, which you're probably familiar with, called the free drug hypothesis. And simply put, that hypothesis indicates that when drugs are consumed by a patient or introduced into their system, the vast majority of that drug is bound by protein in the circulation. And when it's bound by protein, it's pharmacologically inert. So what really matters in terms of a drug's ability to have effect is the fraction of itself that is unbound from protein, which we call that the unbound drug concentration. There are, of course, a variety of other variables in the equation that I've listed for you on the right, but ultimately that single parameter is what governs success. And that single parameter is what we have a very difficult time predicting in preclinical models. So here's an orthotopic animal model of a small molecule, which is a Wee1 inhibitor that was designed by Merck. And you can see in the cross sections, this stippled penetration of the molecule on this multi-mass spec, which is fairly unimpressive. And you can see, at least in this analysis, they looked at the concentration total of the drug, not just unbound in brain and plasma. And it gives you a ratio that's fairly uninspiring. You can represent that ratio as what we call the KP, which in this case was 0.04, which is something that you would assume is inconsistent with the drug having any effect in a brain tumor. However, the same drug went into a clinical trial where the PK and PD were analyzed, and the results were diametrically opposed to what we saw in rodents. And this has been replicated over and over with different drugs. And the fact is that there is a lot of species specificity that we don't fully understand with the blood-brain barrier, where a drug or a therapy can perform one way in a primate or a rodent model or a large animal model like a canine and perform another way in a human. There's been a lot of effort trying to decipher this and predict this and sidestep these problems with more advanced technologies. PET microdosing or radioactive labeling of a molecule is one strategy that's, I think, increasingly common and not quite yet, I would say, at prime time. So this is a drug, AZD1390, that some of you may be familiar with. It's an ATM inhibitor that was designed by AstraZeneca with the express intent of being brain penetrant. They took another drug with a similar backbone and then modified that structure in order to give it the biophysical properties of a penetrant molecule. And then they put this molecule through a PET microdosing study. And with that data, you can generate kind of a mathematical model of what would be the projected KPUU, which is the unbound ratio of drug in brain to plasma. And so a KPUU of 0.24 isn't bad. This same molecule, though, went into a phase 0 hybrid study with us, and I won't belabor the study design, but simply to let you know that we have study designs today that let you measure PK and PD performance. And even though there was a lot of effort put into identifying how this drug would perform in humans, it ended up being about a 10-fold difference in performance, not in a bad direction, but nevertheless in a fairly unpredictable way. So this is all to say that there are other strategies to better interrogate these questions in the context of drug development. And there have been review studies like this one from Mike Vogelbaum that tell us very clearly that we as a field are not engaging these strategies remotely as frequently as we need to. I'll try to delineate some of the nomenclature here so we're all speaking the same language. You're familiar with phase 1, 2, 3 studies. You may be familiar with what are called microdosing phase 0 studies, which is what typically happens in medical oncology, where you give perhaps 1/100th of a dose of a drug. But those studies don't really work typically in blood-brain barrier kind of encased scenarios. So we have the phase 0 title, which is really assigned to studies where a drug without therapeutic intent is administered to a patient in the hours to days before surgery. Then the patient goes to the operating room and tumor PK and tumor PD is assessed by tumor acquisition. So these may be studies where perhaps one pill, one dose of the drug is given, not enough to have any therapeutic intent, but enough to measure PK/PD. A window of opportunity study is where that drug is being administered with therapeutic intent preoperatively, perhaps for days or weeks or months. And then at some point, the patient goes to the operating room, tumor PK/PD is analyzed, and then that patient goes back on the regimen, continuing until progression. So a key differentiator between these two models is therapeutic intent, but it's also what happens after surgery. At the Ivy Center, at our program, we've kind of embarked upon a hybrid version of this, which we call phase 0 hybrid or PK/PD triggered studies. These are phase 0 studies in the sense that there's still a very limited amount of drug administered to patients preoperatively, so there's no therapeutic intent, but that also means there's very little opportunity for risk. But then when the patient goes to the operating room, there's a real-time analysis of tumor PK and PD, and that lets us decide whether the patient should go on drug afterwards or not. This is a kind of a critical decision point because it can save the patients a lot of time as well as risk. If the drug is not demonstrating what is predetermined to be an adequate PK/PD response, then the patient simply pivots to some other experimental therapy without losing any time. And conversely, if there is evidence, then the patient goes on to that drug with some confidence that this may have effect. These types of hybrid phase 0s can happen at any point during the phase 1, 2, 3 sort of continuum, depending on where the drug is in its developmental stage. I mentioned that, from a regulatory perspective, there's no universally accepted definition of a brain penetrant molecule. So the term is used fairly loosely in our literature and in our community. I think we would advocate, and many would as well, that it should be defined on the basis of unbound drug concentration in gadolinium non-enhancing tumor tissue, simply because gadolinium enhancing tumor is really not a major oncological problem for us. It can be easily resected in most cases through surgery. It can be easily targeted with adjuvant therapies. It has a more permissive blood-brain barrier. It's non-enhancing disease that is what is incurable. The operatic study you're probably familiar with, which is a window of opportunity trial of a PARP inhibitor called olaparib. This was combined with temozolomide in recurrent GBM patients, and they attempted to do sort of a PK/PD study looking at total tumor concentrations of drug. Now, one of the challenges is that when you measure the total amounts of drug, but not the unbound, it can give you a different picture of what's happening with respect to the pharmacologically relevant fraction of the drug. In this study, the other challenge was that the investigators in the protocol used tumor fluorescence as the sole determinant of where the tissue was coming from. And for those of us who operate on these patients, like myself, tumor fluorescence is a really valuable biomarker, but doesn't give you exactly the same results as going based on gadolinium enhancement. In fact, one of the reasons it's effective as a extent of resection tool is that it goes beyond the areas of enhancement. Nevertheless, the conclusion from the study was that there were fairly high levels of total drug in these tissue compartments. And so this led to the follow-on, analysis that this is a brain penetrant molecule. Now, the same molecule was analyzed as part of a comparator cohort in a dual drug phase 02 study that we completed in the same patient population. And again, without belaboring the study design, there was an arm in this study where olaparib was administered pre-surgically, and then at one of two time cohorts after the last dose, tumor was acquired, and we analyzed the unbound concentrations of olaparib. Now, only patients that had a predetermined threshold of unbound drug were able to go on to long-term dosing, and that threshold was 5-fold the biochemical IC50 of the drug. So here you see several patients that all received olaparib. The colorized bars demonstrate the unbound concentration of olaparib in their gadolinium non-enhancing tumor, and the horizontal dotted line represents that minimal pharmacological threshold that we determined, which for the case of olaparib was 25 nanomolars. And so you can see that with one exception, most patients didn't come close to this threshold. And in fact, the total amount of unbound drug was only 4 to 7 nanomolars in gadolinium non-enhancing tumor. So we would look at that data and we'd reach the opposite conclusion, that this is not a brain penetrant molecule and probably should not be pursued in the context of a therapeutic study in GBM. And so this kind of emphasizes the importance of pharmacokinetic contextualization. I'll give you another example. This is a small molecule infigratinib that a cohort of investigators used to target various FGFR aberrations, because this is it's a pan-FGFR inhibitor. So you see on the left this waterfall plot showing you changes in tumor volume. And of course, as in many of these studies, there's a subset of patients that have what seem to be positive or desirable changes in tumor volume. And you see on the right the swim lanes, which show individual patients and their clinical performance in the context of infigratinib. And once again, as we always see in uncontrolled phase 2 studies, there's a subset of patients that seem to do well. And so the conclusions drawn from these data are typical of the conclusions we draw from data like this all the time in our field, which is that there must be some subset of patients that are responsive to this drug. And naturally, our job should then be to identify what is special about these patients so that we can select them better for the follow-on study with the same drug. Now, that kind of analysis is making some assumptions that really are not tested in the study, specifically in regards to how the drug is penetrating these patients' tumors. So here's the same drug, and if you focus on the panel on the left, you see these colorized vertical bars that show the unbound concentration of infigratinib in gadolinium non-enhancing tumor, and the horizontal dotted line demonstrating, the desired minimum effect. You can see in patient 1, the concentration is well above that minimum threshold. At patient 2, it's just at it. At patient 3, it's well below. And the top panel shows you the pharmacodynamic effects of those same 3 patients, with patient 1 in orange having an expected pharmacodynamic suppression consequent to FGFR inhibition, patient 2 having a somewhat equivocal effect, and patient 3 actually trending in the wrong direction. And so this kind of shows you this inter-individual heterogeneity, and the fact that not all of these 3 patients should even be considered to be put on this drug, as patient 3 really has no chance of having an effect from the drug, even if the patient does really well. So in fact, 2 of the patients went on the drug per protocol, and they both had early recurrences. And in their case, what we saw was a loss of the target. So the FGFR aberration in patient 1, for example, was FGFR TAC3 fusion. Well, that fusion was selected out in that tumor at recurrence, which tells you that the drug was doing something, but obviously not enough to affect the tumor ecosystem. The other thing that these kind of data show you, again, is that if you look at the gadolinium enhancing component of the tumor, you're going to get a different interpretation. In this case, you would assume that all patients are getting enough drug into the tumor, which is not the case. And finally, if you look at CSF as a surrogate, which is often done in the drug development space, there's really no relationship between CSF drug concentrations and tissue drug concentrations. And we've done at this point well over 500 phase 0 patients for various drugs. We have yet to see any correlation between CSF drug concentrations in the unbound fraction and unbound drug in either gadolinium enhancing or non-enhancing tumor. These are just simply different compartments in the brain. So I just gave you kind of a preview of how you analyze pharmacodynamic effects, looking at PK performance and then PD. I'll speak a little bit more about that in detail for those interested. This is another phase 0 hybrid study. This is a CDK4/6 inhibitor, abemaciclib, combined with an ERK inhibitor, this LY321 molecule, in recurrent GBM. The idea being to kind of hit in a vertical combination 2 targets along the same pathway. So a study like this, as with many of these studies, begins with a screening phase. This is a recurrent glioblastoma study. So these patients have tissue already, either from pretreatment biopsy or from archival. And so there are molecular entry criteria that are logical for these kinds of targeted inhibitors. And then for patients who qualify, they get both drugs for 6 days. There's a what we call time escalation cohort. You see their arm A and B, where the interval from the last drug dose to the time of the craniotomy is controlled and varied. And this is because these drugs have kind of somewhat unpredictable PK performances. It's not entirely easy to extrapolate how they performed in rodents to humans. And so we need to create a little bit of spread in the data points. Ultimately, though, the true litmus test is the non-enhancing unbound fraction of the drug. And in a drug combination, you need to see both of the drugs in excess of the threshold in order to proceed with any confidence. So this emphasizes the complexity of polypharmacy approaches. Nevertheless, when patients meet both those criteria, they then go on to get both drugs in a therapeutic regimen and in a statistically designed phase 2. So in a study like this, the PK analysis is relatively straightforward, but settling on what a PD readout is for each of these molecules can be a little bit complex. So for ERK inhibition, for example, there are probably a half dozen readouts that could be argued as a readout for ERK inhibitor efficacy. We ultimately settled on one, but the point is that this is something that has to be worked through in advance. If we kind of cut to the end in terms of the performance of these drugs, the performance was unremarkable, is probably the best way to put it. And these are the swim lanes for the first 7 patients, and a median PFS of 2.7 months in the recurrent setting is really nothing spectacular. And so this is something we would conclude as a negative study. Now, the question is, why did this combination not work? Well, part of the insight comes from looking at each of the drugs' relative effects. So we know a CDK4/6 inhibitor works through phospho-Rb, and so we can see the relative before and after levels of that. And we know an ERK inhibitor, at least in part, works through phospho-Rsk. And we know that in both cases, we're looking to decrease proliferative indices like MIB1. Now, if we look at phospho-Rb, we see some decline in it. It's statistically significant, and that's really looking at all of the patients in the phase 0 component of the study, not just the ones that go on to the phase 2. If we look at the phospho-Rsk, it's more equivocal, and it's unclear what's happening here, but clearly nothing dramatic. But the balance of effect is this decrease in proliferative indices. However, the reason I show this data is to make the point that it's all about what your comparator tissues are. And in these studies, if you're using tissue from archival specimens, as we often do, there's plenty of genetic drift that happens from the time of initial diagnosis to recurrence, where you can't make assumptions about what the biology of the tumor is at recurrence based on what happened, 6 months prior, before temozolomide and radiotherapy and the effects of resection. So it's really critical for these studies to judge your readout based on pre-treatment biopsied patients. And this means that in our field, we have to be pressing for more biopsies as part of routine standards of care. And there is a movement afoot for that, as many of you know. If we look at the patients, for example, in the phospho-Rb category with pre-treatment biopsies, we see a pretty clear trend for these 3 patients. If you look to the right, however, in phospho-Rsk, that trend is opposite. And in fact, it's very clear that the real failure in this combination is the ability to modulate the phospho-Rsk pathway. Now, pharmacodynamics are not just sitting there static like a mountain, in the vista. These are mobile and dynamic endpoints that change as the tumor evolves. And so this is why for all of our phase 0 studies, we now introduce intracavitary Ommaya reservoirs so that there can be real-time monthly CSF draws from the cavity that give you some indication of where the biology of the tumor is going relative to your experimental therapy. So here you see several different time points for this patient after resection, in the perioperative space, in the long-term sort of recovery space. And all of those things are somewhat independent in terms of data from the radiographic results. So here we see the emergence of an EGFR amplification well in advance of what is radiographic evidence of recurrence. These kinds of canaries in the coal mine can help you plan for what needs to be done to this patient next. Whether EGFR amplification is a true mechanism of resistance to, for example, this patient's experimental therapy was niraparib, which is a PARP inhibitor. Whether PARP inhibition leads to that is unknown. However, it does provide you with something targetable that you can interrogate in the setting of expected failure. And so that's exactly what happened. At 10 weeks, we see the emergence of the EGFR amplification. At 14 weeks, we see the radiographic emergence of progression. The patient is then pivoted onto a new-in-class EGFR inhibitor that looks to be brain penetrant, BDTX-1535. And so there is this seamless transition and this dynamic dosing of experimental therapy from one lily pad to the next, trying to maintain continuous pressure on this patient's tumor as its genetics evolve. So in the last few minutes, I'll sort of put this all together for you. I've shared with you some examples of these drug development techniques that I think need to be more fully embraced in the space. But the question is, how does this move the needle in terms of really getting results, not just in terms of early promising data, but in terms of these phase 3 studies that we started off talking about. So let's look at the drug class of PARP inhibitors as a proof of concept for this. PARP inhibitors are, sort of an increasingly large class of agents that have emerged in recent years, mostly because of data supporting the synthetic lethality of PARP inhibition with BRCA mutant tumors. And so these drugs have been approved for things like ovarian cancer and fallopian cancer because they truly provide a survival benefit through that synthetically lethal mechanism of these BRCA mutant tumors. However, there are other ways in which PARP inhibitors work, and one of them is just by simply destabilizing DNA damage response and making tumors more susceptible to DNA damage. And we now understand very well how that works for this drug class. And it works not just through the direct inhibitory effect of PARP on the molecule itself, what's called the catalytic inhibition of PARP, but also through a secondary effect called allosteric trapping. And what that means is the PARP inhibitor itself not only inhibits the PARP1 molecule from its own enzymatic effects, but when that PARP1 molecule attaches to damaged DNA to repair it, it prevents that molecule from leaving the damaged DNA. And so this is called chromatin retention. And that seems to be actually the dominant mechanistic effect of PARP in the setting of DNA damage response. Now, I show you on the right these various drugs in the class, and this happens in our space all the time, where a drug class emerges, and there's not just one or two drugs, there could be 3, 4, 5 drugs in that class. Now, historically, to be frank, in our space, most centers and most programs and investigators are just simply going to test whatever's available to them. But the point is that can introduce a lot of bias and irregularities that you would want to sidestep if you could. So if we look at the drugs in this class, we can see that many of them on this curve, where the more you are to the left, the worse you are at PARP trapping, are actually living over there. And I'm circling some drugs that you're familiar with, like veliparib. Veliparib, as we know, was the target of a randomized phase 2 study in newly diagnosed GBM in combination with radiotherapy and temozolomide. This is called the VER2 study, and this was read out as a negative study. And then more recently, veliparib was a target of a larger randomized phase 2/3 study by the Alliance that looked at it in the setting of temozolomide alone, and this also read out as negative. Now, the point I'm trying to make is that, in fact, these molecules, even though they're all part of the same class, don't all behave the same way, at least with respect to PARP retention on the damaged chromatin, or what we call PARP trapping. There's clearly differences between the drugs in these classes, and there are other pharmacokinetic differences. So our program, instead of just pursuing whatever is available, decided to just survey the field in terms of not only PARP trapping and pharmacodynamic effects, but importantly, the pharmacokinetic effects of these drugs, which are also different. And so it was quite clear that one drug stood out in terms of PK performance, and that was niraparib, which was best in class. And here I'm showing you the summary PK performance of the unbound concentration of the drug when it's combined with radiotherapy in a newly diagnosed MGMT unmethylated population. And you see on the right the KPUU, which is the unbound ratios, being fairly provocative. But importantly, this was accompanied by pharmacodynamic effects that give you confidence that this drug's PARP trapping is truly translating to something that will give you radiosensitization. And so this drug was selected out of the batch to then advance into this proof of concept phase 02 study in combination with radiotherapy in unmethylated GBM. And I don't need to remind this audience that 60% of newly diagnosed GBM patients are MGMT unmethylated, and those patients are insensitive to temozolomide, even though they all receive it. And so for those patients, outside of surgery, the real only effective adjuvant therapy modality is radiotherapy. And so it stands to reason that if we have a drug that could improve the effects of radiotherapy, that this is the population that warrants it. And so you see on the left the swim lanes at 2 different drug doses for these patients at initial diagnosis. And then on the right, the survival curves that were read out at a recent meeting with a median OS of 20.3 months. Now, this is a small sample size, uncontrolled phase 2. So we all have to understand what we're looking at. The historical median OS for this population is less than 12 months. So this is a substantial increase from that. But all this tells us is that there's a question to be asked. We have a drug that has best-in-class PK/PD performance. That drug in an uncontrolled setting is giving you a substantial increase compared to historical and overall survival. But that doesn't tell you anything until you put it to the test in a randomized study. But the point is that randomized studies are hard to come by in our space because they require incredible investment. And these are the kinds of data that help provide confidence to our biopharma partners that this is where their investment should go. And so GSK, who make niraparib, analyze this data and then reach the conclusion that we would have reached, which is that, in fact, this does warrant asking the question. And so that is asked in the context of a phase 3 prospective randomized study. This is called the GlioFocus trial. The accrual for this study opened in June. It's a global study. It's about 13 countries worldwide, including the UK, 450 patients randomized 1-to-1, with one arm getting the Stoop regimen and the other arm getting the Stoop regimen minus the temozolomide and the niraparib replaced. So it's a head-to-head comparison. Now, we'll see where the data takes us on this, but the point is that this is probably how pivotal studies should be arrived at in our space. And these are the types of data, not just radiographic and clinical, but biological, that should be driving those decisions. And so the take-home points here really are that as valuable as preclinical data are, they really need to be validated in human brain tumor tissue before we get ahead of ourselves. These PK/PD-driven studies not only tell you the biological performance of a drug, but they give it to you in the context of a clinical performance. It's one thing to look at uncontrolled phase 1-2 data and try to read the tea leaves. It's another thing to look at that data knowing at a patient-by-patient level what the PK/PD performance looks like. And that's what we call biological contextualization. And finally, in a space like ours, where the market forces are not favorable for CNS oncology, we need to recognize that and try to equip our partners in industry with more robust and sort of reliable, I guess is the best word, sets of data that they can base their decisions on for advancing their assets, and reminding ourselves that, in fact, negative data is just as valuable for them, because as much as they want to advance their assets, they also want to shut down assets that are unpromising. And so if a drug comes to them and there may be all the promise in the world, but there's absolutely no chance of this drug penetrating, everyone, including their shareholders and stakeholders, are better off decelerating that and reallocating resources to something else. So I will end there and just remind everyone that what I just described is much of it the product of the Ivy Brain Tumor Center at the Barrow Neurological Institute in Phoenix, Arizona, but almost none of it from my effort, and entirely from this team of young clinicians, scientists, investigators that are focused entirely on phase 0 hybrid, PK/PD driven studies, and early as well as late phase drug development. So with that, I'll end here. Thank you for your time and attention, and we can get on to questions.
Thank you very much, Dr. Sanai. I think I speak on behalf of everyone in the audience here. That was such a fascinating and interesting talk. So as promised, I'm going to open up to some Q&A. So for those who would like to speak, put your hands up, or you can put it in the chat and I'll alternate between the two. You can see I've got a very eager Matt, so I will go over to Matt first. You can unmute to.
Thanks a lot for that. I just wanted to ask a kind of a simple practical question. So these patients are going through a lot with surgery, a Maya reservoir insertion, monthly CSF sample. And I wonder how patients take that, how those discussions are, And I'm also curious as to how you managed to fund it.
Yeah. So our general response from patients has been a high degree of enthusiasm. I mean, maybe the best data point for that has been that we're in the state of Arizona. It's a large patient population, and historically, the majority of our patients have come from this state. Since the program started 5 years ago, that trend has sort of inverted, and the majority of our patients now come from out of state. So people are coming because they want to be part of it. That being said, Matt, I think that the other part of getting a patient aligned with the strategy is just simply the physician-patient communication. And what we found is that these dialogues really need to happen very, very early on in the patient's sort of journey. Many times, those dialogues around clinical trials happen after an operation, perhaps when they meet the clinical neuro-oncologist as an inpatient or often as an outpatient. But we found that in trying to foreshadow these kinds of strategies early on, it really is something that you engage the patient in, sometimes through the surgeon alone, oftentimes within a day of knowing that there is even a problem. We have probably 40% of our protocols are newly diagnosed patients. So you can imagine that that means we have to have a dialogue with this patient before they even know what they have, and socialize them to the concept that this could be a glioblastoma, this could be unmethylated. What does unmethylated mean? And here are some strategies that could protect them or hedge their bets a bit in the event that they end up having something like that. But in the end, I think it's just about creating trust with the patient and their family, letting them understand that we're doing this because there are some difficult things that are going to be asked of us. Supporting it, I think we're very fortunate at the Ivy Center to have a high degree of philanthropic support. And it's really philanthropic dollars, unfortunately today, that are the ones with the least strings attached that let you move the fastest. So while we have probably about 15% of our studies or our work supported by, federal or non-federal competitive funding, the majority of our work is funded by philanthropy. And in fact, I think that is an extremely important strategy for investigators like us, where we have federal funding sources that continuously diminish proportionally, and we have limited advocacy for our patient population compared to other patients with more prevalent diseases and with better survival rates.
That's really helpful, and I suspect you would get similar answer in the UK, even though the situations are very different. Thanks.
Thank you. I see Kevin O'Neill's got his hand up. Kevin, would you like to unmute and ask a question?
Hi, Nader.
Hi, Kevin.
Fantastic talk. Thank you so much for taking your time out for that, and a great strategy. I just have a couple of questions. You mentioned that therapeutic threshold. You didn't mention how you got to it, but that was one question. And also, this whole biological contextualization in terms of seeing that free drug level in the non-enhancing area of the brain. I'm assuming that makes an assumption that that's a fairly uniform distribution through non-enhancing brain, but I also assume that it's probably not correct to make that assumption. So I suppose what my point is that how robust is that as a measurement in terms of deciding whether to go or no go? And have you seen
Yeah, great questions. I mean, both questions kind of come down to the larger question of, how much drug is enough, and how much is not, and the short answer is we don't know, for any drug class what that answer is. With respect to the threshold, our feeling philosophically was there had to be something firm. It couldn't be subjective. And it had to be a bar high enough that to the average investigator, you would say, okay, that's a lot of drug. So that's how we arbitrarily arrived at the 5-fold biochemical IC50. Now, maybe it's too high, I don't know. certainly the majority of the studies we do, the drugs don't achieve that. But the only way to tell, the answer is probably different on a drug by drug basis because their pharmacodynamic effects are different. So I don't have a good answer on that. I think that is the best we could do with the limited knowledge that exists in our space for these drugs. With respect to the intratumoral heterogeneity, of course, that's a problem really for any biological parameter of a GBM, whether it's drug or intrinsic, et cetera. What we try to do is be very specific in how we're targeting these regions by predetermining the voxels on preoperative MRI, making sure that we're not inadvertently sampling areas that are gadolinium and non-enhancing, but for example, MR perfusion high. We want to get these fairly quote unquote indolent areas of the tumor where the blood-brain barrier you would expect is more normalized. But yes, there's going to be heterogeneity, and that's probably one of the biggest challenges we have in our field is the more we look, the more heterogeneity there is. We went from, interpatient heterogeneity to intratumoral heterogeneity, and now there are data for intracellular heterogeneity. I mean, I think we try to draw the line somewhere.
Yeah. And a quick question as an extrapolation of that, in terms of getting it right, there are so many things that we try to get right and we can't. But of the studies that are promising and suggested that it might work, so you go phase 0, phase 2, how many have not actually worked? Because you can't tell whether if there's a no-go, whether it might have worked because you're not gone that far. So
yeah. I mean, I feel like in our field, there has to be a little bit of philosophical alignment, and our perspective, simply put, is that things that work are obvious, and things that don't work do not. There's no other field like ours that has things like Raynaud criteria or stuff like that. We have that stuff because nothing works, and we're trying to, split hairs. If you look at the original Caring Cross data on temozolomide, for example, which was basically done in the CT era, I mean, even then it was obvious this drug was doing something. You didn't need to wait for the Stoop study, although you needed to. So
I remember seeing that first report.
And so we kind of subscribe to this philosophy that what we should be looking for is high signal to noise. And when we look at it that way, so far in 5 years, we've done, I don't know, maybe 24 phase 0 studies. There have only been 2 molecules that have probably held up to that standard. One of them is niraparib, which is now in a phase 3. And the other one is AZD1390, which is an ATM inhibitor that's now entering a phase 2/3. And in both cases, our group got that molecule and we showed best-in-class PK/PD. And, not surprisingly, the uncontrolled clinical data is encouraging, and you have to, put the molecule to the test. So I think that the failure rate should be high because it's a difficult disease.
Yeah. Thank you very much.
Thank you. Ola, would you like to unmute and go? And then Matt.
Hi, Nader. Really fantastic talk. Really interesting. Thank you. I had a couple of questions. It's really impressive to see sort of your pre-treatment biopsies. What is your current practice around that? Do you offer pre-treatment biopsies to all patients, including newly diagnosed and recurrent, or is it just for the newly diagnosed? And also, do you have any advice for centers that sort of might want to think about implementing that, but it's not part of their usual practice?
Yeah, great question. So we certainly do it a lot more than most centers. And here's the logic for it. Number one, in the recurrent setting, I think that as a field, the pendulum has just swung way too far towards advanced imaging. And we all sit in these tumor board clinics and we scrutinize SPECT data, perfusion data, gadolinium enhancing data, and we play this parlor game in trying to guess what it is, when really you can get a lot of clarity in just sticking a needle in this thing, and you're probably saving a lot of money, and you're probably saving the patient a lot of angst. Now, in the upfront setting, I do think that there are a couple things that need to be course corrected in our field, and many of this may be region specific. But number one, a glioblastoma is not a surgical emergency. You don't need to take the patient to the operating room 24 hours after they have a seizure and end up in your emergency room or urgent care. So there is time to let things settle and figure out what to do. And so when you have a tumor like that, number one, there is always a differential diagnosis. In an older patient, it could be lymphoma. In a younger patient, it could be abscess, et cetera, demyelinating disease. So there is a sound medical rationale for tissue acquisition. But then on top of that, from a patient perspective, what we found is that, a patient who was healthy on Monday, in his mind, has a seizure on Tuesday, and you're queuing him up for a craniotomy on Wednesday, is really being put through some incredible stress that perhaps isn't necessary. We found that for many patients, that patient that comes to you with a seizure and you don't know exactly what they have, but you're worried it's a glioblastoma, putting them through a needle biopsy gives everyone a chance to kind of take stock, plan the care in a calm, controlled way, and yes, create an opportunity for an experimental therapy for what is otherwise an incurable disease. Now, the way we've implemented it to make this all feasible within the US system is, number one, we do these needle biopsies as an outpatient. So they come in the morning, they have 30 minutes of general anesthesia. The procedure itself literally takes 7 minutes. Needle in, needle out. They wake up, they get a head CT, and they go home. The complication rate is less than 1%. This is nothing we invented. The first data on this came from Mark Bernstein at the Western in Ontario. This is nothing on the cutting edge, but it lets people get out of the hospital instead of waiting there, and it's cost efficient. In the recurrent setting, that's particularly effective. So this hasn't been a problem for us in terms of the payers of our system. And I actually suspect that in some ways, perhaps not politically, but from a system standpoint, it could be easier to deploy in Europe, where you have more of a single payer system and you don't have insurance companies with each their own standards. That being said, in our US system, we've never had a patient not rejected, I should say, by the payers in the US system for this. So that's been our strategy. Now, there is a movement afoot, like US and internationally, to try to pull that pendulum back towards the middle and make the case for these needle biopsies. I just think that it's good clinical care. And I think that any neurosurgeon and clinical and neuro-oncologist that advocates for it is sitting on very, very firm ground.
Well, thank you. That's really helpful. Lily, am I allowed to ask a second question, or are the other people waiting?
As long as Matt's now put his hand down, so I will let you go instead.
So, final question for me. I was just curious, what are your thoughts about AstraZeneca's reportedly blood-brain barrier penetrant PARP inhibitor, the AZD9574, and also how you think that will impact the ongoing GlioFocus trial?
Yeah, so that's the PARP1 selective molecule, right? So, the data I've seen on it looks consistent with what you'd expect. I think that the concept of PARP1 selectivity is really an issue of, toxicity management, because the hypothesis is the PARP2 inhibition is what's leading to some of the marrow-related and other complications. But here's the difference. So, the short answer is I don't think it's necessary in our space if you're looking at PARP inhibition. But here's a real paradigm shift of how we have to think about drugs. We're not in the chemotherapy era anymore, right? And so all of these small molecules, more of the drug does not mean more biological or clinical effect. In fact, for many of these molecules, it's a Gaussian curve, and more of the drug at some point gives you less biological effect. So in the case of the niraparib drug, for example, the typical dosing for that could be 300 or 400 milligrams. I think the MTD is 400 milligrams. But what we found is that there is no added PK/PD benefit in going from 100 to 200 to 300 to 400. In fact, anything at 100 or more gives you the same basic performance. So, so number one, you can sidestep toxicity just simply by not maxing the patient out with some chemotherapy strategy to MTD. And so we talk at the Ivy Center not about MTD, but OBD, which we call optimal biological dosing, which means you just titrate the dose to where the best PK/PD performance is. And most times that's not the MTD. So if you look at it from that perspective, yeah, a PARP1 selective inhibitor is nice because you could max it out with more safety. But I think at least for what we want to use these drugs for, which is not synthetic lethality, but this radiosensitizer approach, maxing it out isn't the real strategy anyway. So, you know, that's kind of how I read the development of that asset.
Thank you. That's really interesting.
Okay, thank you. Matt, do you want to do the final question before?
I'll be really quick. So, Nader, you had some really interesting talk. Have you looked at doing window studies combining drug and radiotherapy?
Do you mean like the radiotherapy happens before surgery?
Yeah, even a single fraction. So, for example, if you think that PARP is a radiosensitizer, then it might make more sense to explore PARP plus radiotherapy in the setting.
We tried it. So we tried it two different ways. One was the single fraction concept, and the other one was to do it ex vivo, where the radiation happens when the tissue's out. So what it lets you do in the setting of PARP is it lets you see the immediate effects of the radiation and how PARP inhibition modulates that. So that really has to do with the PAR levels. But radiosensitization itself is not just about PAR levels, of course. It's really about longer-term DNA damage leading to apoptosis. And so, you know, unfortunately, the time scale for that happening is sort of inconsistent with a surgical time scale. Our read is that you need kind of many more days to truly see the in vivo radiosensitization effects compared to PAR levels, especially if you're looking at like the accumulation of double-stranded DNA breaks, the consequent apoptosis and cell death from that. So we just haven't found an easy way, Matt, to incorporate it into like a reasonable clinical workflow. And that's been the case with other strategies as well. So for example, like there are some drugs where you do have to give them for 2 or 3 weeks in order to reach steady state or even approximate steady state. And, you know, you can't delay someone's surgery for 2 or 3 weeks. And then there are other drugs where it's totally unclear when you should be looking for a pharmacodynamic effect. So for example, like, I don't think there's any consensus on checkpoint inhibitors. What is the right time to look for, you know, the immune consequent effects of a checkpoint inhibitor in a tumor? Is it 3 days after, 3 weeks, 3 months? Like, I don't think we know. So certainly the model is sort of incomplete in terms of how to apply it to all of these paradigms, and radiosensitization is one of them. The best we could do was to say, look, we see the pharmacodynamic effects that we think should lead to radiosensitization, but the real proof of radiosensitization will be in the phase 3. Now, that being said, for MGMT unmethylated tumors, the rate of pseudoprogression is far less than 50%. It's something closer to like 20-30% in newly diagnosed patients because nothing works. In our phase 02, it was something closer to 60-70%. So, you know, that gave us some hope that that's what we're seeing.
That's really interesting, and there's a conversation I'll pick up there offline around somewhere.
I'm always here.
Thank you once again for giving up your morning/afternoon, and thank you to all the audience who have joined. I think everyone would like to say a big thank you to you. And as always, these sessions are recorded and they will be made available on the YouTube channel, which Matt has kindly put into the chat. And our next Horizons in Neuro-Oncology will be on the 18th of February. Once again, the link is in the chat for that, and you can register as always by Eventbrite, and we'll be welcoming the Head of Research and Development at Care Bio and developing a new functional precision medicine. So thank you once again.
Thank you, everyone.
Thanks
Have a
very
good
much.
evening.
Thank you very
Thank you.
much.
Bye.
Thank you.